1922, Uppsala, Sweden. A Sami child stands stripped bare for measurement, positioned in front of a caliper and a camera at the newly founded State Institute for Racial Biology. The institute's founder, Herman Lundborg, sent teams north for years, examining and photographing Sami families, sorting them into a racial hierarchy built on nothing but skin, bone, and assumption. The stated goal wasn't curiosity, it was proof. Proof that the Sami were a separate, lesser branch of humanity, one that didn't belong in the Nordic story Lundborg and his colleagues were determined to write. 100 years later, geneticists finally ran the actual DNA. What came back was an answer nobody standing in that Uppsala room in 1922 could have predicted, and certainly not one they wanted. The real Sami genome carries a signature tracing back to Siberia, a thread that shows up nowhere else in the racial narrative those early scientists were so intent on constructing. Everything they measured with calipers, everything they photographed and filed away as evidence, missed the one thing that actually mattered, because it wasn't written on the surface of a body. It was written in molecules they had no way of reading. Consider what it means that the tool of oppression and the tool of truth turned out to be the same instrument, separated only by a century of technology. The racial biologists thought they were building a permanent record. Instead, they built an archive of everything DNA would later prove wrong. Stick around, because this one has a genuinely strange twist buried in the middle of it. Hit subscribe if you like these deep dives into the science hiding underneath history's ugliest chapters. Before any of that genetic twist can make sense, the story has to start with the people themselves, not the scientists who measured them, not the theories built around them, but the Sami as they actually were, living at the edge of Europe long before anyone arrived with a caliper. Their history, their geography, and their languages all set the stage for what the DNA would eventually reveal. And that starting point begins not in a laboratory, but on the land itself. Across the vast Arctic region the Sami have called home for thousands of years. The Sami are the indigenous people of Sapmi, a homeland stretching across northern Norway, Sweden, Finland, and into Russia's Kola Peninsula. Today their population is generally estimated in the tens of thousands, spread across all four of those countries. Reindeer herding is the image most people picture when they think of Sami life, and it remains the most iconic tradition associated with them. Though it was never universal, plenty of Sami communities built their lives around entirely different economies. Distinct dialect and language communities exist across the Sami region, and historically these divisions limited intermarriage between groups, keeping populations from different parts of Sapmi genetically and culturally distinct from one another for generations. Coastal Sami built their lives around fishing and the sea. Forest Sami followed a different rhythm entirely, tied to woodland resources. Mountain and reindeer Sami followed yet another path, centered on herding across high, open terrain. 3 different economies, 3 different relationships with the land, all under the single umbrella of Sami, a detail that becomes important later, once the genetics enter the picture. The Sami hold the distinction of being Europe's northernmost indigenous people, a population that has lived at the edge of habitability since the ice sheets of the last glacial period finally retreated and opened the north to human life. Surviving in that environment for thousands of years required an entirely different set of adaptations than the populations living further south in temperate Europe. Then there's the language. Sami belongs to the Uralic language family, the same family that includes Finnish and Hungarian. That places it in a completely different linguistic category from the Indo-European languages spoken by nearly every population surrounding Sapmi: Norwegian, Swedish, Russian. A Uralic language sitting in the middle of an Indo-European Scandinavian landscape is not a small detail. It's a linguistic island, isolated in a sea of unrelated tongues. That mismatch is the first real clue that something about Sami origins doesn't fit neatly into the traditional story of ancient, unbroken Nordic ancestry. Language alone can't prove where a people came from, but it raises the exact question that would eventually send geneticists looking for an answer written in DNA instead of vocabulary. Long before DNA entered the conversation, 19th century Scandinavian scholars had already split into 2 competing camps about who the Sami actually were. One camp framed them as a primitive, ancient Aboriginal population, present in the north since time immemorial. The other camp framed them as foreign Asiatic intruders, outsiders who had moved into Scandinavian territory rather than originating there. Both frameworks claimed to explain the same people, and both arrived at opposite conclusions. There was even a third thread running through the debate. One competing concept held that the Sami were actually an ancestral Nordic population that had once inhabited the entire Scandinavian region, later pushed north by other groups. 3 theories, 3 different origin stories, and not one of them backed by anything beyond observation and assumption. None of this was academic curiosity for its own sake. Both major theories were put to political use. The ancient Aboriginal framing was used to argue the Sami had no special claim to land rights, since they were simply one Nordic population among many. The foreign intruder framing was used to justify treating the Sami as outsiders in the very homeland they had occupied for millennia. Either way, the Sami lost. Skull shape, hair color, and stature became the accepted forms of evidence, treated as hard scientific fact for decades before anyone had the tools to test a single gene. Measurements taken with calipers and rulers stood in for what should have been genetic data, and entire policies were built on those measurements. The rise of racial biology as an institutional force gained particular traction in both Sweden and Norway after the dissolution of their political union, a moment when defining a separate, lesser racial category for the Sami served a broader project of hardening ideas about Nordic identity itself. It gave the movement a political urgency it might not otherwise have had. None of these frameworks, whichever side of the debate they fell on, had any actual DNA behind them. They were built entirely on external measurement, cultural assumption, and the political needs of the moment. The real test of either theory wouldn't arrive for another 100 years, and when it finally did, it wouldn't come from a caliper. It would come from a lab. The first real test of Sami origins arrived through the work of Günther and colleagues, published in PLOS Biology in 2018. Their team sequenced 7 Scandinavian hunter-gatherer genomes, some at coverage as high as 57x, dating from roughly 9,500 to 6,000 years before the present. This was the deepest genetic look yet into the population that first colonized the North. Scandinavia was one of the last regions of Europe to become habitable after the last glacial maximum, and until this study, the migration routes of its earliest colonizers had remained unclear. Nobody knew exactly how people first reached this frozen edge of the continent once the ice began to retreat. What the genetic data revealed was an east-west gradient running in the opposite direction from the pattern seen everywhere else in Mesolithic Europe. Instead of one clean wave of settlers, the picture was more complicated, and it pointed toward 2 entirely separate migrations converging on the same territory. One group moved up from the south. A second, later group arrived from the northeast, following the ice-free Norwegian Atlantic coast northward. These 2 founding populations met and mixed within Scandinavia itself, producing hunter-gatherers who were genetically more diverse than their counterparts in Central and Western Europe at the same point in time. Instead of a single ancestral line, Scandinavia's earliest inhabitants were already a blend, shaped by 2 different points of origin folding into one population. The same research team identified elevated frequencies of light-skin pigmentation variants in this ancient population, along with a signal in the gene TMEM131, possibly tied to long-term adaptation to cold environments. Both findings fit a population that had been living at the northern edge of the habitable world for thousands of years. This mixed hunter-gatherer population forms the deep genetic foundation beneath everything that would come later in the north. But it is not yet the Siberian layer that makes the Sami genome so distinctive. That signal belongs to a different chapter of the story, one written along an entirely different line of descent. The paternal side of Sami ancestry was mapped out through the work of Tambets and colleagues in 2004, which established that Sami Y-DNA is dominated by 3 haplogroups, N1C, I1, and R1A. Of these 3, N1C is the most common, with I1 following closely behind as a near-equal second. Within the Sami population specifically, researchers identified a distinct subclade of N1C, labeled N1C-L1025, a variant that carries particular significance for tracing paternal origin. I1, interestingly, is also the most common haplogroup found in Sweden as a whole, and when researchers examined the Jokkmokk Sami population specifically, they found a structure for I1 and N1C strikingly similar to that of neighboring Swedish and Finnish populations. On the surface, that similarity might suggest deep, uninterrupted connection to the surrounding Nordic world. But N1C itself tells a different story. This haplogroup traces back toward Siberian and Uralic-speaking populations to the east, a paternal thread that does not originate in Scandinavia at all. It is a lineage that entered the region from somewhere else entirely, carried westward over an unknown span of generations, until it became one of the dominant paternal signatures among the Sami today. Alongside this eastern signal, researchers also documented something else, The Sami show markedly lower Y chromosome diversity than the populations surrounding them. Low diversity of this kind is not a sign of continuous mixing with neighboring groups. It is a signature of long genetic isolation, a population that largely bred within itself across many generations rather than blending outward with its neighbors the way I1 frequencies alone might suggest. Put together, the paternal record paints a population with two forces working against each other. One line, I1, sits comfortably within the broader Scandinavian genetic landscape. The other, N1C, points firmly east into Siberia, a thread with no real precedent in the Nordic world. One line runs toward the neighbors next door. The maternal side of the story runs in a very different direction entirely. Two mtDNA haplogroups, U5b1b1 and V, together account for almost 90% of the entire Sami maternal gene pool. That figure alone sets the Sami apart from nearly every other population in Europe, where maternal lineages tend to be far more scattered. U5 itself is one of the oldest mtDNA lineages known on the continent, identified in human remains more than 30,000 years old, placing its origins deep in the Ice Age rather than anywhere near the modern era. Within that lineage, U5b1b1 alone exceeds 40% frequency in some Sami subgroups, an extraordinary concentration by any European standard. Most populations show far more maternal diversity than that. This particular branch likely originated in an Ice Age refuge population, one of the pockets of humanity that survived the worst of the glacial period, and it expanded northward as the ice sheets eventually retreated and opened new territory. A detailed phylogeographic study focused specifically on what researchers call the Sami motif within U5b1b1, examined the lineage across 31 different populations. That research traced its likely origin to Western Europe rather than the East, anchoring the maternal side of Sami ancestry firmly in the same refuge populations that repopulated the continent after the ice retreated. Set that beside what the paternal line showed. The maternal line is almost entirely ancient European in origin, while the paternal line carries a signature that traces back toward Siberia. That asymmetry between the two sides of Sami ancestry is not a minor technical detail. It is a finding worth sitting with, because it means the two halves of the Sami genetic story point toward two completely different points on the map. Two lineages, two different continents of origin, fused together into a single population over thousands of years of shared history. Mothers carrying ancient European ancestry, fathers carrying a lineage rooted far to the east, both threads converging in the same small population at the edge of the Arctic. And that convergence is only the beginning of the picture, because a smaller, more specific Siberian signal runs through the rest of the Sami genome as well, one that hadn't entered the story yet. Autosomal analysis, the kind that looks across the whole genome rather than just the maternal or paternal lines, puts the Sami's Asian-linked genetic component at roughly 5 to 8%, with one study estimating it as high as 13%. That range is small in absolute terms, but it is a real and measurable signal, distinct from the broader European ancestry that dominates the rest of the genome. Some Sami individuals carry the mtDNA haplogroup D5, specifically the subtypes D5A3A1A or D5E. This same haplogroup turns up among Norwegian Sami, Finns, ethnic Russians, and the Mansi people of western Siberia, linking the Sami to a wider network of populations stretching east. D5A3A1A itself descends from the broader D5A3 lineage, a lineage also documented in Tibet, Japan, and Korea, with the root of haplogroup D as a whole tracing back to western Siberia. A second Siberian-linked lineage, haplogroup Z, appears in roughly 7% of Sami sampled from Finland, adding another thread to the eastern side of the genetic picture. Not every marker tells the same story, though. Haplogroup H1, found among the Sami, was virtually absent in a sample of 781 Samoyed and Ob-Ugric Siberians, yet present in Western and Central European populations. That absence matters. It shows that the Siberian and European genetic layers did not blend evenly across every marker in the Sami genome. Some lineages moved freely between the two worlds. Others stayed confined to one side or the other. The Siberian signal itself carries a twist that runs against the obvious assumption. It isn't a recent trickle, a small addition arriving in the last few centuries and slowly diluting outward. The oldest ancient samples tested actually carry more of that Siberian ancestry than living Sami do today. The proportion has gone down over time, not up. That single detail upends the tidy assumption that Siberian ancestry arrived recently and spread forward into the present. It suggests something closer to the opposite. A concentrated Eastern signal present early, gradually diluted across generations of mixing with neighboring populations. That reversal deserves a much closer look, because it changes the entire timeline of how Siberian ancestry actually entered the Sami genetic record. Lamnidis and colleagues, publishing in Nature Communications in 2018, sequenced 11 ancient individuals recovered from the Kola Peninsula and from Finland, the first ancient DNA ever recovered from mainland Finnish soil. 8 of those individuals came from the Kola Peninsula, including 6 from a single burial roughly 3,500 years old, while the remaining 2 came from an 18th to 19th century Sami cemetery, giving researchers both a deep ancient sample and a more recent historical one to compare against each other. Within that Kola Peninsula population, researchers identified the earliest known evidence of Siberian ancestry anywhere in Fennoscandia, dating to around 4,000 years ago. That eastern signal, present in the region by that point, later spread from the Kola Peninsula into Finland itself, carried forward through subsequent generations. The team's central surprise came from comparing old against new. The oldest samples in the entire study carried the highest proportion of Siberian ancestry of any individuals tested, ancient or modern, higher than anyone living in the region today. Specific individuals from that Kola burial carried Siberian mitochondrial haplogroups Z1, C4, and D4, and 2 of the men carried the Y chromosome haplotype N1C1A1A, also written as NL392, a direct paternal link to the Eastern lineage running through the broader Sami genetic story. Put together, these results describe a genetic history that didn't build gradually toward the present the way a simple ancestral line would suggest. The Siberian ancestry that defines the Sami today started out concentrated, present in high proportion 4,000 years ago, and thinned out steadily across 3,500 years of continuous mixing with the populations living around them. That timeline forces a correction to the obvious assumption. The purest ancient sample recovered from that Kola burial was not simply the untouched ancestor of today's Sami, passed down in a clean, direct line. It was already a population in transition, already changing, long before its descendants ever became the Sami communities that exist now. The ancestry didn't flow forward unchanged. It diluted, generation after generation, on its way to the present. The same 2018 study turned its attention to Löwenluhta, a lake burial site in South Ostrobothnia, Finland, roughly 1,500 years old and located far south of where any Sami community lives today. Researchers analyzed the genomes recovered from that Iron Age site and found something that didn't match the modern map of Sapmi at all. Those Iron Age individuals showed greater genetic affinity to modern Sami populations than to modern Finnish speakers living in that same region today. In other words, the people buried at Löwenluhta, more than 1,000 years ago, were genetically closer to today's Sami than to the Finns who now occupy that very land. This finding adds physical, genetic evidence to something the ancient DNA record had already begun to suggest: that populations genetically similar to today's Sami once occupied territory well south of where Sami communities currently live. The modern boundaries of Sapmi, spanning northern Norway, Sweden, Finland, and the Kola Peninsula, represent a contraction of that historical footprint, not the original extent of it. Several forces likely drove that contraction over the following centuries. Language shift, the expansion of agriculture into territory better suited to farming than herding, and later Scandinavian settlement all likely pushed the genetic and cultural footprint of Sami-related populations steadily northward, generation after generation, until it settled into the boundaries recognized today. This single find complicates the old racial biology framing that treated the Sami as a fixed, isolated Arctic population, one that had supposedly always occupied only the far north and nowhere else. That framing depended on the Sami being unchanging and static across time. The Löwenluhta burial evidence says otherwise. A population that once stretched into the lake country of South Ostrobothnia, hundreds of kilometers south of the modern Sami homeland, does not fit inside the box the 1922 institute in Uppsala built for them, with calipers and photographs. The land itself moved under the Sami long before anyone tried to measure who they were. The westward spread of early Neo-Siberian ancestry may be directly tied to the dispersal of the Uralic language family, the same family that includes Sami, Finnish, and Hungarian among its living branches. That connection links a genetic movement to a linguistic one, and the two appear to trace remarkably similar paths across the map. Neo-Siberian populations diverged from broader East Asian populations more than 11,000 years ago, then expanded outward into Siberia itself, where they mixed with and gradually replaced earlier Paleo-Siberian groups already living in the region. That expansion set the stage for the Eastern genetic signal that would eventually reach as far west as Scandinavia. The linguistic mismatch raised earlier in this documentary, a Uralic language sitting isolated among Indo-European neighbors across Scandinavia, now has a genetic explanation running alongside the cultural one. It is no longer just an oddity of vocabulary and grammar. It has a population history behind it. This does not mean genes and language always move together as a single, inseparable package across every region they touched. But in this case, the timing of the genetic spread and the direction it traveled line up closely enough with the Uralic language dispersal to be more than simple coincidence. The Uralic-speaking world stretches an enormous distance, from Hungary in the west all the way to Siberia in the east, and the Sami sit at its extreme northwestern edge, about as far from the language family's origin point as it is possible to be while still belonging to it. The ancient genetic layer recovered from sites like the Kola Peninsula and the language family the Sami still speak today both point back toward the same general eastern homeland. They arrived separately, through different mechanisms and different populations, but moving in the same rough direction, over roughly the same span of centuries. With that deep, shared ancestry now mapped across both genes and language, the next question turns inward, toward how unevenly that ancestry actually landed across the Sami's own internal regional divisions, from the coast to the forest to the mountains. North Sami, the largest of all the Sami subgroups, shows genetic affinities closer to Finns and Scandinavians than to some of their own more distant Sami relatives. That alone should complicate any assumption that Sami describes one uniform genetic population. Skolt Sami, traditionally based in northeastern Finland and Russia, tell a different story entirely, showing stronger genetic links to ancient Siberian populations than any other Sami group tested. Linguistic and cultural boundaries between these communities historically limited intermarriage, and that separation reinforced the genetic distinctions between groups generation after generation. Even within a single country, the pattern holds. In Sweden, the Jokkmokk Sami show a Y-DNA structure closer to neighboring Swedes and Finns than to some other Sami communities living under the same national flag. None of this tracks cleanly onto the modern political map of Sapmi, the four-country boundary spanning Norway, Sweden, Finland, and Russia's Kola Peninsula. Skolt Sami sit genetically closer to Siberia than North Sami do, regardless of which country's border happens to run through their territory today. Political geography and genetic geography are simply not telling the same story. A single averaged Sami genome, the kind of simplified figure that gets quoted in a headline, hides all of this internal structure. The reality is a mosaic, a set of related but genetically distinguishable communities, each shaped by its own history of migration, isolation, and contact with different neighbors. Coastal economies, forest economies, and reindeer herding economies didn't just produce different ways of life. They produced different genetic signatures, preserved across centuries of limited contact between groups. That mosaic did not survive by accident. Something specific kept these distinct communities from simply dissolving into their much larger surrounding populations over the thousands of years they lived alongside Scandinavians, Finns, and Russians. Whatever that mechanism was, it operated consistently enough and for long enough to leave a genetic signature that scientists can still read today, group by group, almost like a fingerprint of isolation itself. Genome-wide analyses place the Sami in their own distinct cluster, separate from other European populations entirely. On a genetic map built from populations across the continent, the Sami don't blend smoothly into their neighbors. They stand apart as a recognizable, self-contained group. That separation shows up clearly in the details. The Sami display lower mtDNA diversity than other European populations, a pattern suggesting their maternal lineage descends from a relatively small number of founding lines that persisted, largely unchanged, across thousands of years. The same pattern appears on the paternal side. Sami Y chromosome diversity runs lower than that of the populations surrounding them, reinforcing the same picture from a completely different angle. Long-term reproductive isolation, sustained across generations. Geography did much of this work. Arctic terrain, low population density, and a subsistence economy built around reindeer herding all limited gene flow between Sami communities and outsiders for centuries. Simply put, there weren't many opportunities and little incentive for large-scale mixing with populations living in more hospitable regions further south. Cultural practice reinforced whatever geography had already established. Intermarriage patterns and community boundaries did the rest of the work, especially between the coastal, forest, and reindeer herding branches of Sami society described earlier. Each of those economic divisions functioned almost like its own semi-closed system, marrying largely within itself. Geneticists recognize this as a textbook founder effect signature. It is not evidence of some pure or unchanged population frozen in time, the kind of framing racial theorists once reached for. It is simply evidence of a population that mixed internally, among its own branches and communities, far more than it mixed externally with the much larger population surrounding Sapmi on every side. That isolation, real and measurable as it was, became something else entirely once outsiders got hold of it. What geneticists today read as an ordinary founder effect, born of geography and subsistence economics, later got twisted into a very different and far more damaging story, one told not in a lab but from a pulpit, and later from behind a caliper. Long before any racial theorizing entered the picture, the Sami were first compelled to submit to Swedish law under Charles IX. In that early period, they were quietly permitted to continue practicing their own religion in private, a limited tolerance that would not last. By the second half of the 17th century, Swedish authorities moved to force Sami communities to abandon their traditional shamanic practice entirely. The private tolerance of the earlier era gave way to open suppression. Sami drums, central to traditional ritual and divination, were seized and burned wherever authorities could find them. Joik singing, the traditional vocal tradition central to Sami cultural expression, was banned outright. Sami communities were forced to submit to church doctrine, not only in public life but within their own private and family practices as well. The Noaidi, the Sami spiritual practitioners who used the drum for divination and ritual, became the primary target of this campaign. Their role was treated by church authorities as a direct rival to religious power, a competing source of spiritual authority that could not be allowed to continue alongside official church doctrine. This campaign of religious suppression predates any formal racial biology theorizing by more than 2 centuries. The pressure to erase Sami distinctiveness did not begin in a laboratory or an institute. It began in the church, with confiscated drums and banned songs, long before it was ever dressed up in the language of science. Very few of the original Sami drums survive today. Most of what scholars now understand about their symbolism and use comes from a small number of confiscated examples that happen to be preserved rather than destroyed, fragments of a much larger tradition that was otherwise systematically dismantled. Religious suppression laid the groundwork for everything that followed. 2 centuries later, a different institution picked up the same project of erasing Sami distinctiveness, this time working not through scripture and confiscation, but through calipers, cameras, and the language of race. After the political union between Sweden and Norway, which had lasted from 1814 to 1905, finally dissolved, racial biology gained significant traction in both newly separated countries. In the wake of that dissolution, the Sami began to be formally categorized as a distinct racial entity, a classification that carried consequences far beyond academic debate. This new racial classification pushed the Sami toward exclusion from the broader Nordic population in the public imagination. Where the Sami had once been understood, however imperfectly, as part of the Nordic world, they were now being redefined as something categorically separate from it. The policy framework that grew out of this racial concept portrayed the Sami as a biologically weak and primitive population, one supposedly requiring state protection. In practice, those so-called protective measures primarily benefited the state itself rather than the Sami they claimed to serve, bringing Sami land under direct state control and opening the natural resources on that land to extraction by outside interests. This is the exact ideological soil the 1922 Institute in Uppsala would eventually grow out of. It was not an isolated project, dreamed up independently by one ambitious founder. It was the culmination of decades of hardening racial categorization, building steadily in both Sweden and Norway following the dissolution of their union. The theories being tested under this framework were never neutral science conducted for its own sake. They were built, deliberately, to justify a specific set of land and resource decisions that were already underway, giving a scientific veneer to policy choices the state had already made. In Uppsala, that decades-long theorizing would finally turn into something formal. A state institution with a name, a founder, and an official government mandate. Sweden's State Institute for Racial Biology was formally founded in Uppsala in 1922, under the direction of Herman Lundborg. Researchers connected to the institute traveled extensively through northern Sweden, forcing Sami, men, women, and children, through degrading physical examinations, cranial measurements, and nude photography, all conducted in the name of scientific classification. The stated purpose of this work was to classify the Sami as an inferior race, one whose culture was framed as fundamentally incompatible with modern European progress. Under an accompanying state policy known as Lapp ska vara Lapp, meaning the Lapp shall remain a Lapp, the Swedish state segregated nomadic reindeer-herding Sami from the rest of society. Standard educational opportunities were denied to Sami children under this policy, built on the assumption that they simply didn't need the kind of education offered to other Swedish citizens. This was not fringe pseudoscience operating on the margins of respectable society. It was a state-funded institute, backed by government authority, running continuously for years with official sanction. The measurements collected inside its walls were treated as legitimate biological data at the time, and that data was used in real time to justify segregationist education policy affecting entire Sami communities. Across the border, a parallel campaign was running through an entirely different mechanism. Where Sweden had built an institute focused on measurement and classification, Norway's campaign aimed less at documenting Sami difference and more at erasing it altogether, working not through calipers and cameras but through the machinery of the school system itself. Norwegianization, known in Norwegian as fornorsking, drew heavily on social Darwinism and rising nationalism, framing Sami culture as primitive and uncivilized, a way of life that needed to submit fully to the authority of the Norwegian nation-state. This wasn't a vague cultural preference. It became formal government policy, backed by law. The Land Act of 1902 decreed that state land could only be sold to people fluent in Norwegian who had also adopted a Norwegian name. In practical terms, this stripped many traditional Sami herders and fishermen of ancestral land their families had managed for generations, converting them overnight into tenants on territory that had once been theirs outright. Years earlier, in 1899, the Växelsenplakaten forbade Sami people and Quens from working as educators in multilingual schools. That single decree marked the start of a full assimilation policy explicitly aimed at reducing minority group identity and weakening community cohesion from within. The residential boarding school system became the primary battleground for this cultural erasure, the place where Sami children were systematically separated from their language, their families, and their traditional way of life, generation after generation. The reach of this project extended even beyond the living. Sami graves were opened and skeletons removed for study, extending the racial science project into the dead as well, treating even Sami ancestors as raw material for a classification system built to diminish their descendants. The policy's explicit racial justification lost much of its force after World War II, as openly racial language became politically untenable across Europe. But the underlying project of assimilation was not abandoned. It was simply reframed, repackaged as regional social development rather than the racial and cultural erasure it had always been. Policies built to erase a distinct Sami identity ran for well over a century, reshaping land ownership, education, and family structure across generations. Their aftershocks did not stay contained within Scandinavia. They reached all the way across the Atlantic, to a different continent entirely, where their consequences are still being counted today. An estimated 30,000 people of Sami descent now live in North America, a population built largely through emigration during the 19th and 20th centuries. Many of these emigrants made a deliberate choice upon arrival. They concealed their Sami identity specifically to evade the systemic racism and forced assimilation pressure they were leaving behind in Scandinavia. Sami women in particular experienced a compounded form of discrimination during this period. Eugenic ideology, colonialism, nationalism, and gendered oppression all overlapped within the same set of policies, layering multiple forms of pressure onto Sami women specifically, in ways that shaped both their choices and their descendants' inherited relationship to that history. Scholars studying this North American diaspora describe the resulting identity concealment as a direct contributor to intergenerational trauma, shaping how Sami identity is experienced and understood among descendants living in North America today. Grandchildren and great-grandchildren often grew up with almost no knowledge of their own ancestry, a silence inherited from choices made out of necessity generations earlier. One Sami woman, quoted in a 2001 Norwegian newspaper interview, described the scale of that erasure in blunt terms. It took 100 years to Norwegianize the coast Sami, and it may take another 100 years to make them Sami again. That single statement captures both the depth of the assimilation project and the slow, uncertain work of reversing it. These migration numbers rarely appear alongside the DNA headlines that get the most public attention, but they are not a separate story. They are a direct downstream consequence of the exact same policies that produced the racial science examined earlier. Proof that the damage done in Scandinavian institutes and schoolhouses didn't stay confined to Scandinavia at all. Decades later, one of the institutions most responsible for that history would finally put a name to it in public. The Church of Sweden formally apologized in 2022 for its history of anti-Sami persecution, a public acknowledgement centuries in the making. That apology does not stand alone. It sits inside a much longer arc, stretching from 17th century drum burnings and banned joik singing through 20th century racial biology and its state-sanctioned measurements, all the way to this 21st century public reckoning. No equivalent formal apology has closed the loop on every institution involved in that history. The State Racial Institute's records and photographs, the same cranial measurements and nude photographs taken from Sami, men, women, and children, still exist in Swedish archives today, a physical remnant of a project that was never fully accounted for by the state that funded it. The DNA evidence gathered a century later did not validate a single one of the racial categories that institute was built to prove. Every hierarchy Herman Lundborg's team set out to document simply wasn't there in the genetic record, no matter how the data was examined. Modern genetics instead found something the eugenicists were never looking for and, given their aims, could never have used anyway: a genuinely mixed Siberian European ancestry, built through migration and gradual blending over thousands of years, not a fixed racial hierarchy of the kind those researchers spent careers trying to establish. The science that was supposed to prove Sami inferiority ended up documenting something closer to the opposite. It revealed an Arctic population that survived repeated, sustained attempts at religious suppression, forced assimilation through education and biological classification, and remained genetically and culturally distinct despite every one of those efforts. Which brings the story back around to the one question the 1922 Institute in Uppsala set out to answer, and never actually got right. The verdict, arriving a century late, is that Sami ancestry is a genuine hybrid. A maternal line dominated by ancient European lineages, U5B1B1 and V, fused over thousands of years with a paternal line carrying a Siberian-linked haplogroup, N1C, producing a population built from two very different points of origin, rather than one unbroken source. The Siberian-linked autosomal component sits around 5 to 8%, occasionally estimated as high as 13% in some analyses. That signal is real, measurable, and genuinely present in the Sami genome. But it is a minority signal, not the majority ancestry that older racial theories once imagined defined the entire population. That Siberian layer arrived in stages, beginning at least 3,500 to 4,000 years ago, and entering the region via the Kola Peninsula, rather than sweeping in through a single dramatic invasion. It built gradually, layer by layer, across multiple generations of contact and mixing. Isolation, not purity, explains the low genetic diversity documented throughout this story. A small founding population mixed heavily within itself and only rarely with outside groups, producing the reduced diversity geneticists observe today. That pattern reflects geography and subsistence economics, not any inherent biological separateness. Regional Sami communities, North, Skolt, and others, carry genuinely different proportions of this shared ancestry, meaning there is no single representative Sami genome that could stand in for the whole population. The internal mosaic documented earlier turns out to be central to the story, not a footnote to it. None of this maps onto either camp from the 19th century debate, not the ancient pure Nordic theory, not the foreign inferior race theory. Both were wrong, built entirely on measurements and assumptions that could never see what actually mattered. Buried in DNA nobody in that era had the tools to read. Return to the 1922 examination room. The calipers, the camera, the underlying assumption that biology would ultimately prove a hierarchy, ranking the Sami somewhere beneath the surrounding Nordic populations those researchers considered their own. The actual biology, decoded a full century later, proves something closer to the opposite. It documented resilience through mixture, not inferiority through isolation. A population shaped by two continents of ancestry, rather than diminished by either one. The ancient DNA record even rewrote the map itself, showing that Sami-related populations once lived far south of where Sami communities live today, in territory that now sits at the heart of central Finland. The boundaries drawn by history did not match the boundaries drawn by biology. The Uralic language puzzle, the regional genetic splits between North and Skolt Sami and every community in between, the founder effect isolation shaped by geography and economy rather than race. Every one of these threads traces back to real migrations and real historical circumstances, not the racial categories that were built afterward to explain them. Some of the story remains genuinely unresolved. Researchers do not yet agree on exactly how tightly the spread of the Uralic language tracked alongside the Siberian ancestry wave, or on precisely how many separate migration pulses actually reached Sapmi over the millennia. The full picture is still being filled in, 100 years after a state institute tried to measure the Sami into a hierarchy. The real question left standing isn't what their ancestry proves about rank at all. It's why that question ever needed answering in the first place.